All right, guys, let's go ahead and get started here. So today we're kind of at this transition point between the skeletal muscle and the nervous system. Okay, we'll have a couple more classes on skeletal muscle, but this idea of the neuromuscular junction is really this transition between the nervous system and the muscle, skeletal muscle, and where the nervous system interacts with skeletal muscle to engage it. Okay, so we're going to talk about the neuromuscular junction here. And again, we've already talked about the things that happen after the neuromuscular junction gets activated. We talked about excitation contraction coupling, which is getting ready for contraction. We talked about cross-bridge cycling, which is actually the contraction part. Now we're kind of taking one step backwards and looking at how did we start excitation contraction coupling. That's really where we are here. Okay, so there's three parts to the neuromuscular junction. Okay, so the presynaptic terminal, this is the end of the motor neuron. Okay, so this is the nervous system side. So motor neuron is a neuron that is sending the signal out from the central nervous system out to the periphery and controlling some kind of tissue outwards, also known as an efferent neuron. We'll talk about that a lot more coming up. Okay. This presynaptic terminal has a bunch of little vesicles in it that contains neurotransmitters, and the one for skeletal muscle is called acetylcholine. And I'll just abbreviate it as ACH most of the time when you see it on the notes. Okay, so that's the nervous system side. And then in between the two, we have just a space, right? And that's called the synaptic cleft. Okay, so it's the space between the axon and the muscle cell. This is the diffusion space for that neurotransmitter. The neuron's going to release the neurotransmitter, it's going to diffuse through there, and then make its way over to the skeletal muscle. Okay, this space contains an enzyme called acetylcholine esterase, and this is the enzyme that helps us break down acetylcholine after it's been released. Okay, and this is going to help us by leading to relaxation. Okay, if there was nothing to break down acetylcholine, then it would lead to contraction for a lot longer than we really want it to. Right? And so we have an enzyme in place to help us break that down and help us relax muscle when we're done contracting it. Okay, and then lastly, the muscle side of the neuromuscular junction is the motor end plate. We already talked about this part before. It's just a specialized region of the muscle cell membrane, the sarcolemma, heavily folded, so we can have a lot of surface area, so we can put a lot of these receptors for acetylcholine in the membrane. Okay, one thing to note about these receptors is they're also ion channels, right? And there's something we'll talk about later that's called a ligand-gated ion channel. Right? So ion channels are things that allow ions to pass across a membrane. And ligand is just like a chemical messenger of some sort. It attaches to that protein that's the receptor. And when it does, it's like a key. It opens up the gate. Right? It opens up that gate for ions to come in or come out of the cell. Okay. All right. So take a second. Identify those different parts to it here with your team, and then go ahead and send in the answer to this one, and it's going to be one of the letters, okay? Go ahead. So you're sending the letter of whatever's pointing to those, wherever the acetylcholine would bind. What's that special structure that the acetylcholine would bind to? Send in the letter of that. Give you about 10 more seconds. All right, we got 59 so far, 61. Probably a few more of you need to send that in. 63, 65, good. That might be close. All right, I think that's probably everybody, 65. All right, let's see. See what you guys said. Okay, so the majority of you, 78%, are saying C. Okay, and what would that be called? What would we call C? Okay, motor end plate. Some of you are saying, what's another guess? That's kind of the whole structure, but what specifically is where acetylcholine binds to? The receptor, right? Yeah, acetylcholine receptor, for sure. Okay, so when it comes to those numbers up there, we're just naming those. So what do we call one? Yeah, so that presynaptic terminal, all right? Okay, and then what do we call two? Yeah, the synaptic cleft. Okay, and then what do we call three? You guys said it before, but what is it? Yeah, the motor end plate. All right, and then with the letters here, so we see A, it's a little vesicle full of neurotransmitters, so that's one of those acetylcholine vesicles. Okay. B is pointing to just a little blue dot. Wouldn't necessarily know what it is, but that would be an example of acetylcholine esterase. We find it both bound to the membrane on the muscle side and also floating in the synaptic cleft. We find it in both places. Okay. C, you guys already said, was acetylcholine receptor. Okay. This is just calling it a transmitter gated channel. Okay. Acetylcholine is a neurotransmitter. And the neurotransmitter is what opens or closes the gate of that channel to allow ions to flow. And we'll talk about what those ions are in a second. Okay. And then what do we got next? D there. D is called a voltage-gated sodium channel. That's not one that's on the previous slide. We're going to talk about those in a bit. But when we talk about an action potential is really the signal that the nervous system sends. We can also have action potentials on muscle cells and other electrically excitable cells. We have to have those special voltage-gated sodium channels in order to have an action potential. Okay, so we'll talk a lot more about those in the next couple of weeks. Okay, and then we've got E, which is a different kind of channel, also not on your previous slide, but another important voltage-gated channel. This one specifically allows calcium to come in. And we will talk about that one here in just a second and how it functions in what we're doing here. And then finally, F. What is F there? What would F be? Yeah, that's our neurotransmitters. That's acetylcholine. Okay, so we got acetylcholine in these little vesicles. Somehow this is going to get the neuron is going to get stimulated to release those acetylcholine vesicles into the cleft. They're going to go across, bind to the acetylcholine receptor, lead to a signal, and we'll talk about what that is in a second. Okay, any questions about just the kind of the components of the neuromuscular junction there? Okay. All right, so let's talk a little bit about, before we get into talking about what an action potential is, we first of all need to just talk about kind of general electrical properties of a cell. Okay, and so when we look at a cell, this is representing a cell membrane right here. Okay, on the right side of this is the inside of the cell. On the left side of this is the outside of the cell. Okay, and you can just tell by looking at those ion concentrations there that one of those ions is we have much more of it on the inside of the cell, and there's another ion we have much more on the outside of the cell. All right, what's the big one we see on the inside? potassium, right? We got high potassium inside of our cells. Okay, potassium is the primary ion of the intracellular space in the body. Okay, that's where most of our potassium is. It's intracellular. Okay, and then just the opposite for sodium. We have lots and lots of sodium outside of our cells. Sodium is a primary ion of the extracellular space. Okay, so that's just part of the normal makeup of kind of a resting state of a cell. Okay, but then on the inside, we also have lots of these high negatively charged proteins. Okay, those are represented by these A's with the negatives on them. Okay, yeah, and that's kind of the bottom line there. And so what this results in is the inside of the membrane being much more negatively charged, or the inside of the cell being much more negatively charged than the outside of the cell. Okay, so positive out here, negative out here. Okay, and there's a couple reasons why that is the case, and we'll talk about those on the next slide. But the key from this here is understand that we got lots of potassium on the inside, lots of sodium on the outside, lots of negatively charged proteins on the inside. And that's our first thing here. Okay. All right. Then secondly, if we look at a cell like this, one of the key things we find on most cells in the body are special little proteins like this that they use ATP, and when they use ATP, they pump 3 sodium ions out of the cell, and they pump 2 potassium ions into the cell. Okay, this is just known as a sodium-potassium pump. Okay, and so our normal resting membrane potential is about -70 millivolts. What that means is -70. We have a charge of -70 on the inside of the cell versus the outside of the cell. Okay. Yeah, and so what I'm going to do, we'll come back to the importance of the sodium-potassium pump here in a second, but I'm going to show a video that kind of walks us through resting membrane potential and why it is the way it is. So let me switch over to my computer for that for 1 sec. Okay, here we go. Should be some sound. Some more time. I think Zoom's using my audio. I think that's why it's not working. All right, guys. Well, let's go back to here. Unfortunately, no video. What I will do is I'll walk us through the 3 things that dictate resting membrane potential. Okay, which is what it was going to show us. Number 1, I'm just going to write it here, so I think you'll be able to see that. Yeah, number 1 will be that sodium-potassium pump. Okay, the sodium-potassium pump. When it pumps 3 sodiums out and 2 potassium is in. So the net charge change every time that happens is -1 for the inside of the cell, right? Because we're losing 3 positively charged ions and we're gaining 2 positively charged ions for the inside of the cell. Okay, so the net change is -1. Right, so that's part of what contributes to resting membrane potential. Okay, number 2, which the video showed so nicely, is that there's more leakage of potassium than sodium. Okay, and so when we're talking about leakage, we mean ions are going to move in the direction of high concentration to low concentration, right? And so since there's a lot of sodium on the outside, not much on the inside, if sodium was able to leak, which way would it go? Into the cell or out? It would go in. Yeah, absolutely. It would go in. Okay. And then since there's a lot of potassium on the inside, not much on the outside, if we let it leak, it's going to leak out. Okay. And so there's special leak channels in cells that allow for some sodium to leak and for some potassium to leak. Okay. And there's way more of those potassium leak channels than there are sodium leak channels. So what that means is we're losing more of a positively charged ion to the inside of that, or out of the inside of that cell, much more than we are gaining a positively charged ion with sodium. All right, so if there's more leakage of potassium than sodium, then this is also a net negative on the cell electrical charge. Right, because we're losing more of a positive than we are gaining that positive. Okay, and then third, the one that we already said, is just these negatively charged proteins inside the cell. Okay, so when we put all those 3 things together, that results in the cell being negatively charged. Okay, so when a cell's at rest, when it's not being activated by the nervous system, or if it's the nervous system cell itself, when it's not being activated, we have that -70 millivolts of a charge on the inside of the cell. Okay. That wasn't as exciting as the video, but there you have it. Questions on that? So those are those kind of 3 things that dictate this resting membrane potential. Okay, so yeah, let's see how much of let's see how well you guys are tracking here. We'll go ahead and answer this clicker question. Oops, let me. Okay, so talk about this one with the people on your team. Make sure you're on the same page. Okay, so put in all the answers that are true. If there's more than one, make sure you hit send with this type of question. All right, I got 55 of you, maybe about, I think there's 10 more that need to hit enter or hit send. All right, anybody else? Make sure you hit send with this type of question. Okay, good. There we go. Now we got everybody. Okay, so what do you guys say? So about 2/3 of you are saying A and C, and then about 20% of you are also adding E to that. Okay, so A would be the fact that there's more passive leakage of potassium than there's sodium. Okay, that's true. Okay, and then so obviously B is not true. And then C, the fact that the sodium potassium pump moves 3 sodium out and only 2 potassium in, making it a net negative on the cell resting membrane potential for sure. Okay, and then the E one, the fact that sodium leaks in and potassium leaks out. Yeah, I mean, that's part of it for sure. Yeah, they're both the same charge, so it's not like super crucial, but that's definitely part of it. We just say, when we say one leaks more than the other, it is important to know which way that it's leaking. Right? So if it's leaking, yeah, if we're losing more of a positive charge to the inside, then that's going to make the inside more negative. Yeah. Sodium leaks in or out? Oh, did I not read that right? Yeah, sodium leaks in and potassium leaks out. That's true. Yeah. Okay. Yeah, because there's a lot of sodium on the outside. And so that means sodium's going to want to leak in. Right? It's going to go with its diffusion gradient from high to low. Same thing with potassium. There's a lot of potassium on the inside. It's going to go with its diffusion gradient, try to leak out. Okay. Other questions on that? Go ahead. Yeah, so we're just talking about simple diffusion here, right? So simple diffusion would say that an ion is going to flow, or any substance is going to flow from an area where it has a high concentration to an area where it has a lower concentration. So since we have a lot of sodium on the outside of the cell, not much on the inside, if it was able to leak, it would leak in. Potassium is just the opposite. We have lots on the inside, not much on the outside. So if it's able to leak, it leaks out. Yeah, go ahead. Yeah, not even facilitated, but active transport. Yeah, because that sodium potassium pump is an example of primary active transport where it uses ATP to push both of those against their concentration gradient. So thank you for clarifying that. Yeah. Yeah, for sure. It's yeah, but with the sodium potassium pump, we're moving sodium out, right? And potassium, we're moving potassium in. So it's the opposite direction there. Yeah. And we'll talk about another example later when we talk about action potentials. The sodium rushes in through a different type of channel as well. So there's multiple channel types. But yeah. Okay. Other questions on that? Okay. All right, so just real briefly about an action potential. We'll talk more about action potentials later, but the definition of an action potential is this rapid, reversible, spreadable, which means it propagates down the axon or down the cell, change in membrane potential in the positive direction. Okay, so we go from being negatively charged down here to then all of a sudden shooting way up in the positive direction. That's called depolarization. Okay. And what that does is that spreads down an axon towards the target tissue. Okay, and yeah, and then we'll come back to this more, talk about the details of what happens to cause this action potential. But just for now, know of it as the signal that's just spreading rapidly down either a muscle cell or a neuron. Okay, go ahead, Olivia. Is that the one that is like an all or nothing? Yeah, there is an all or nothing component to that, which we'll talk about more when we get there too. Yeah. Okay. All right. So this was another video that I'm assuming the audio won't work on either. But this just kind of walks us through these steps we have on this slide right here. Okay. And so those videos were directly from Mastering A&P. So you can definitely hop onto Mastering A&P yourself, go to the study area. I think it's called A&P Flicks. It's like these 3D animations, and they're helpful. So you can hop back on and see both the resting membrane potential one and the events at the neuromuscular junction one. But these are kind of the steps involved in the events of the neuromuscular junction. And so I'll just kind of walk through them a little bit. And then what I'll have you do is the next 1, 2, 3, 4 slides that kind of represents those pictures. And so after I kind of talk through them, just kind of want you to look at those pictures, kind of figure out what's going on in each of those, match them up with the steps on the slide. Okay, so this all starts with that action potential coming down the motor neuron towards the muscle cell. Okay, it hits the neuromuscular junction. The first thing that happens there is if we go back to that neuromuscular junction slide, we saw that we had these voltage gated calcium channels. Okay, so those voltage gated calcium channels, they get opened up when that change in voltage or the action potential hits them, right? So that's why we get calcium going into the neurons, because we open up those voltage gated calcium channels. Okay, so calcium goes in, it binds to the vesicles that contain acetylcholine, and then those vesicles are stimulated to go to the surface of that neuron and dump acetylcholine into the space, into the synaptic cleft. Okay, acetylcholine diffuses across, binds to those acetylcholine receptors, and then what happens is acetylcholine receptors are channels as well, right? And acetylcholine opens them up. That causes the gate to open, and now sodium, since there's a lot of it on the outside of the cell, if there's an opening for it, it's going to rush into the cell, and so sodium rushes into the cell, and that stimulates an action potential to then happen on the muscle cell. Okay. And then now we're back to where we started with excitation contraction coupling, right? That action potential goes down the T tubules, activates the sarcoplasmic reticulum, releases calcium, that whole process, and then back to cross bridge cycle. Okay. And then as far as the neuromuscular junction goes, acetylcholine is going to get broken down by acetylcholine esterase. And then eventually that's going to stop the action potentials on the muscle when we move into relaxation. Okay, so take a second, a couple minutes with the people around you, walk through these slides, write in what's happening on the next 4 slides, and then we'll come back together here in a second. Yeah, and you can go ahead and answer this clicker question on after all of those at the end, after you've walked through it, okay? All right, so after you've walked through all those steps, you can go ahead and ask questions.
Oh, that's so it'd be me.
charge. We can also say the membrane depolarizing. So we got 51 of you responded. And you guys are pretty much split half and half down the middle between B and D. So what that tells me is you guys got to turn to each other and convince the people on your team of your answer. And just so you can vote again. All right, we need, I think, 2 more people to vote. I've got 63 on there. Anybody else? There we go. Someone just threw an A in. Figured they'd throw something in. Okay. All right, we're still somewhat split. All right, we'll come back to that in a sec. Okay, so this first step here. So what's happening on this first slide? We've got an action potential, just call it AP, or abbreviated as an action potential, coming down the motor neuron, and it's going to do what? It's going to open these calcium channels on the neuron, right? And so then calcium is going to flow into the neuron. Okay, not the same step as calcium flowing into the muscle cell. That's going to happen later, right? Calcium is one of the primary signaling ions through lots of cellular physiology. So it's just, we see 2 different spots that it signals something here. It signals something in skeletal muscle. It's signaling something here in the neurons. And what's it causing to happen here? Okay, acetylcholine to be released, at least. We'll say that so that calcium is going to cause ACH release. Okay, so we got that. So then acetylcholine is released, and we do see it going down there and binding to the receptors. Okay, so ACH binds to receptors. Okay, and you can see that the same protein that allows it to bind also is an ion channel, right? So that's why it's a ligand-gated ion channel. The ligand is the chemical messenger, which is the neurotransmitter. You can see that it also allows potassium to move, but there's such a stronger gradient for sodium to rush in than there is for potassium to go out, that overwhelmingly more sodium comes into the cell than potassium does go out. And so we depolarize the muscle cell. Okay, and if we do enough of that depolarization, it can spread to the regular part of the muscle cell, and then that can start an action potential. Okay, so we start an action potential on the muscle cell, right? And then this picks up where we left off, or where we started off with excitation contraction coupling, right? So now that action potential propagates down the membrane. It's going to go specifically down these T tubules. Right, it's going to activate the sarcoplasmic reticulum, cause it to release calcium into those sarcomeres. It's going to go bind to troponin, allow the myosin head to bind to actin, and we do that whole cross-bridge cycling thing. Okay. All right, so action potential leads to calcium release in muscle. Okay. All right, questions on any of that so far? Okay, so then this signal or this stimulation will be terminated by the effects of that acetylcholine esterase. I'm not going to have room there. Esterase breaks down acetylcholine. And then we get relaxation of the muscle. That's how the muscle can relax. That's how the signal stops being transmitted. Okay. All right. So any questions about those steps there? Okay. All right. So then with this comprehension question, so 2/3 of you said B, and then 1/3 of you said D. So definitely sodium rushing into the cell is the primary cause of depolarization. Okay, the sodium potassium pump is not related to action potentials happening. It's related to maintaining resting membrane potential. It's also related to recovering from an action potential later, but it's pumping sodium out, right? The depolarization is actually caused by sodium coming in, right? Sodium potassium pump is sending potassium the other, or yeah, potassium and sodium the opposite directions of their normal diffusion gradient. Okay, any questions on that? Okay, just a few clinical applications to the neuromuscular junction. One of these is an autoimmune disorder called myasthenia gravis. This is a disorder where your immune system starts destroying those acetylcholine receptors in your neuromuscular junctions. Okay? You can see the little antibodies binding to those acetylcholine receptors, destroying them. This leads to paralysis, weakness, all things related to not being able to contract muscle. Right, so it's a devastating autoimmune disorder, myasthenia gravis. Okay, tetanus. This is the thing that we have in our normal vaccine regimen when we're little kids. Is part of the tetanus vaccine. Okay, it is a bacterial infection, and the bacteria actually live in the soil. But some of that soil tends to get up into rusty metals, right? And so if you kind of get cut with a rusty metal, or you just get a cut and some dirt gets into it, you have potential of getting this bacteria into you. And what happens is that bacteria releases acetylcholine receptor agonists. Okay, and what that means, an agonist is something that does the same thing that acetylcholine would do. And so it binds to those acetylcholine receptors and just makes your muscle contract like crazy. And there's no special enzyme there to stop it, like there is acetylcholine esterase. And so your muscle just keeps contracting, so much so to the point that you can actually break your own bones with your muscle contraction. It can actually rip tendons right off of bones. It can break bones in half. And it can potentially be life-threatening because if your diaphragm seizes up, then you can't breathe, right? And your diaphragm is skeletal muscle as well. And so it can be life-threatening in that case. Okay. And then lastly, curare is a poison that's actually found on those little poisonous frogs in the jungles of South America. Okay, these are the ones that literally, if you just touch their skin, you absorb that poison and it can kill you. It's that lethal of a poison. And what happens is, so this is acetylcholine receptor antagonist. So that means it binds to those acetylcholine receptors, but it doesn't have the same effect as acetylcholine. It doesn't do anything when it binds to it, except for the fact that it gets in the way of acetylcholine binding to it. So then you can't contract muscle at all. And so again, if this gets into your diaphragm, or I'll come back to that in a sec. If this gets into your diaphragm, you can't breathe anymore, right? You suffocate. All right. And so this is actually part of the cocktail that is used in carrying out the death penalty and lethal injection. Is this drug, a synthetic version of this drug that you definitely don't have to know that name, but just knowing that it's part of that lethal cocktail. That's curare. But it was actually just first discovered on those frogs, and now they make it synthetically. Okay, questions about any of those? All right, guys, have a great rest of your day. We'll see you on Friday. Me and Betsy will be here ready to go. Thank you. All right, see ya.